Welcome back, ladies and gentlemen. So before we jump into the process of meiosis, let's do a real quick recap of mitosis because a lot of the same terminology is going to come back, very similar processes are going to come back with just key differences. So remember when we talked about mitosis, the general overall cell cycle, right? Every cell starts an interphase, and this is going to be the same both if we're talking about mitosis or meiosis. And that interphase, remember, gets split up into that G1 phase, the S phase, and the G2. So G1 being sort of that normal growth and function. If the cells are given the go-ahead to pass that G1 checkpoint, we'll go into the S phase, which is where the DNA will duplicate so that we have enough copies to send to those new cells. And then we'll go into that G2 checkpoint, or excuse me, that G2 phase, where we'll do the organelle duplication and just the final growth and preparations for the division of that cell. And then again, there is a checkpoint at the end of that G2 to ensure that all of that preparation has happened appropriately. Now, if we're going into mitosis, then after G2, we enter that mitotic phase, that cell division phase, which is where we talked about mitosis versus cytokinesis. So, mitosis being the division of that nucleus, and we talked through prophase. Chromosomes are condensing, they're starting to attach to spindle fibers, the nuclear membrane's disappearing. Metaphase, where all of those chromosomes line up along that metaphase plate in the middle of the cell. Anaphase, the sister chromatids split and start moving towards the poles along those spindle fibers. And then telophase, where everything starts relaxing, going back to chromatin, the nuclear membrane's coming back, the spindle fibers have disappeared. And then that overlaps with cytokinesis, where we actually divide the cytoplasm itself and create those two new daughter cells. So those terms, prophase, metaphase, anaphase, telophase, are going to come back. We're going to talk sister chromatids again. We're going to talk spindle fibers. A lot of the same processes are going to repeat in meiosis with a few key differences. Um, when we talk about mitosis, right, the end of just typical cell division we've been talking about, we've been creating two genetically identical daughter cells. And if we're assuming this is happening in human cells, then it's a diploid cell to a diploid cell, right? So if I go through mitosis, they're genetically identical. So if I were to look at this original parent cell, this would be a 2N cell, and if we're talking humans, 46 chromosomes. The two cells that result from that would also be 2N cells with 46 chromosomes each. Okay. We're going to see when we go through meiosis that that's not going to necessarily be the case. Because remember, mitosis in humans, at least, is there for growth and for repair, right? If I need to get bigger, I can't just make bigger cells forever because I start depreciating the efficiency of moving materials across that cell membrane. So I make more cells instead of bigger cells. And if I have damaged cells and I need to replace them, then I do that via mitosis. We get to meiosis, and now I'm going to take those diploid cells and reduce the chromosome number, right? So I'm going to go from a diploid adult, right, which is what we're seeing here, right, that 46 chromosomes, and I'm going to go through meiosis now and reduce the chromosome number in half. So that each of those gametes, whether it's the egg or the sperm, depending on if you're male or female, each of those gametes has only half the number. They're haploid cells, right? And that way, when we come back together via fertilization and we create that diploid zygote again, which eventually becomes the embryo and the baby and the next generation of adults, that we're consistently keeping the chromosome number the same, right? We're conserving that chromosome number is a way to put it. So this is only going to happen in germ cells in adult humans. Those diploid adult germ cells will go through meiosis, cut the chromosome number in half to create these haploid daughter cells, these haploid gametes. And this is going to only take place again in those germ cells that exist in your reproductive organs. All right, so for females, it's our ovaries, for males, it's in your testes. And we're doing this as a result of meiosis In order to do that, though, it's going to now require us to go through 2 rounds of cell division. So we're going to go through meiosis 1, which includes prophase 1, metaphase 1, anaphase 1, telophase 1, and then go through another set of meiosis, meiosis 2, which now is going to consist of prophase 2, metaphase 2, anaphase 2, telophase 2. And that's where some of those differences between meiosis and mitosis start taking shape. 2 rounds of division, and now I'm going to go from 46 chromosomes down to 4 cells that each contain 23 chromosomes instead. So again, the real purpose here is to ensure that I'm not duplicating the chromosome number every time. So I'm going to start off with that diploid cell, and again, if we're talking humans, that means that there's 46 chromosomes in that cell, right? So that diploid number here would be 46. And if you take note, by the time I already get through that first round of cell division, I already have haploid cells, right? Remember, that's what that N means. So there's only 23 chromosomes in these cells by this point. Now the sister chromatids are still attached, so we'll see that. So that's where that second round of cell division occurs, so that each of these cells has 23 chromosomes apiece, so that when I take those eggs or those sperm and put them back together to make that offspring, I recreate that diploid 46 chromosomes and consistently keep that number every generation. So how do we do it? All right, well, let's talk through these phases. So here is meiosis 1, right? Here I'm going to take each of these steps. Now, let me take a step back here for a second and talk about the fact that interphase still exists before this. All right, so interphase is still happening before we go through this round of division. Okay, so we're still going to go through that G1, the S, and the G2. Okay, so we're still going to have some growth. We're still going to have the duplication of DNA. We're still going to prep and make some new organelles and ensure everything is accurate. There are still checkpoints here. Okay. Okay, so all of that stuff is still happening just like we talked about in mitosis. But once we pass into meiosis 1, now we start to see some differences. So I get to prophase 1, all right. Nuclear membrane's still going to disappear. The centrioles, are still moving to opposite sides of the poles. They've been duplicated back in interphase. Spindle fibers are still there. But we're going to see that there's some really distinct differences when we start talking about prophase 1, and that is the fact that now I have to consider homologous chromosomes. Now I'm going to take into account the chromosomes that came from mom and the chromosomes that came from dad. When we talked about mitosis, we didn't really bother mentioning that because those individual chromosomes just duplicated, they all lined up, everything split, and we were done. When we get to meiosis, though, here, now I'm going to separate out those homologous chromosomes. All right, so we're going to see in prophase 1 that those homologous chromosomes are going to come together. They're going to do some mixing of genetic information, and they're going to stay together and line up together in metaphase 1. All right, so metaphase 1, when we looked at, or excuse me, when we just talked regular metaphase, we would have had a purple chromosome, a green chromosome, a red chromosome, a blue chromosome. They all would have been lined up one on top of the other, and we would have split those sister chromatids. Here now, I'm concerning myself with those homologous pairs. All right, it's going to be the homologous pair that lines up. All right, so here's a big difference. We're actually going to call this a tetrad. And if you think about it, tetra is 4, So I've got four chromatids, one, two, three, four. So those homologous chromosomes now are going to be what lines up along the metaphase plate. These tetrads are going to line up, all right? And it's going to be those homologous pairs now that separate, okay? So if I look at this, sister chromatids stay together. Okay, but it's the homologous pairs that separate, right? So if you look at this, right, homologous pairs or homologous chromosomes are what separate from each other and start moving to those opposite sides, okay? So that by the time I get to the end of telophase 1 and cytokinesis, these cells are now haploid. And the reason they're haploid is because they only contain one set of chromosomes. Okay. Why do they only contain one set? Well, I started back here with 23 pairs of chromosomes, right? Pairs of chromosomes, 46 total. I separate out those pairs, and now when I get to the end of the first round of cell division, those pairs are no longer in the same cell. All right, so I've got one set here, one set over here. So prophase 1 is probably, or I should say, meiosis 1 as a whole, really, is where we see the biggest differences between mitosis and meiosis. So prophase 1, right, one of the biggest things that happens here is this process called synapse, where I'm going to take those homologous chromosomes, right, all of them. So chromosome 1 from mom, chromosome 1 from dad, come together in pairs. Chromosome 2 from mom, chromosome 2 from dad, come together in pairs. Chromosome 3 from your mother and chromosome 3 from your dad come together in pairs in your germ cells. And they form these tetrads, right? So here's that tetrad structure, right? Four chromatids all lined up together. They still are cinched at the waist, just like those sister chromatids were before creating that centromere, and they're all held together. Now, the really cool thing about this tetrad is that it's going to allow this process called crossing over to occur. And we'll talk a little bit more about what that means later, but essentially we're going to be able to mix up genetic information from what your mom gave you and what your dad gave you so that when you go to pass that information on to your offspring, we've now mixed up the genetic information that you got before you pass it on. We're creating more genetic variation. And that genetic variation is huge when we talk about why sexual reproduction is such a big deal from an evolutionary standpoint. Remember that evolution is driven by genetic variation. The more variation there is in a population, the more stable it is, the more healthy it is, should an environment change, right? The environment's stable, we can do okay without all that variation. But if everybody's identical to each other and that change takes place, there's no other options for you to adapt and survive as a species. And so the species is way more likely to go extinct as a result of that. So sexual reproduction has lots of places in which this mixing up of genetic information takes place. And one of those things is here in prophase 1 with this process called crossing over. So other components to meiosis 1, right? I mentioned those tetrads lining up in the metaphase plate, right? Those homologous chromosomes are now sitting in that center of the cell as opposed to just being lined up one on top of the other. So rather than seeing 46 chromosomes lined up across that metaphase plate, we now see 23 pairs of chromosomes lined up. We see 23 tetrads lined up on that metaphase plate. Anaphase 1, the sister chromatids remain attached, but the homologous pairs separate, they move to the poles, and then when we get to telophase 1 and cytokinesis, each of the cells that ends up resulting at the end of that are those haploid daughter cells with a total of 46 chromatids. Let's kind of change that Rather than saying chromosomes, just because that can get a little confusing perhaps, let's cross this out and call this chromatids. All right, so 46 chromatids, but there's only 23 chromosomes. All right, so 46 chromatids because those sister chromatids are still attached. 23 duplicated chromosomes, right? One set because they're haploid, right? Okay. Um, Other things that are still happening here, right? Spindle fibers are shortening during anaphase 1. We're pulling those chromatids towards the poles. Telophase 1, the nuclear membrane still comes back and reforms, at least temporarily. The genetic information relaxes again. So a lot of those sort of underlying components that are happening in mitosis are happening here too, right? So we go back right here, that nuclear membrane's coming back, spindle fibers have shortened, we've only got a set of centrioles in each. Anaphase 1, they're moving along those spindle fibers towards the poles, everything lines up in the metaphase plate. In meiosis, just like mitosis, it's just the difference of how they're lined up. And prophase 1, again, the nuclear membrane still had to disappear, spindle fibers and centrioles still needed to move. So all of that underlying stuff is still happening. It's just what's really happening to the chromosomes here that's different. All right. Then we get into meiosis 2, and now this is going to look a lot more like mitosis. The only difference here is that I'm working with half the genetic information because these are all haploid cells, right? And I've got two cells simultaneously going through this process at once. So I talk prophase 2 now. All right, there is a really short little interphase here, but no duplication of information. Okay, so really short interphase, but no gene duplication, right? No DNA duplication. So there's no S phase. Okay, that's really important. I don't want to duplicate the information again here because that's kind of counterproductive. I already have my sister chromatids here. I don't need to make more of them. So the whole point to meiosis 2 really is the division of those sister chromatids. So we go through prophase 2 in each of those individual cells I created at the end of meiosis 1. Centrioles still need to duplicate, move to the poles, nuclear membrane still needs to get out of the way. And now when I get to metaphase 2, I've got 23 chromosomes lined up here, 23 chromosomes lined up here along those metaphase plates. And now the sister chromatids are what splits here in anaphase 2, right? Okay, so those sister chromatids are now pulled apart from each other. Okay, so that they each end up into individual cells. I go through telophase 2 now, cytokinesis as well. All right, nuclear membrane comes back, and the chromosomes relax back into chromatin, and now I have 4 haploid daughter cells that are all genetically different from each other and genetically different from the original parent cells that we started with, not only in the sense that they are haploids, they have half the genetic information, but also in the arrangement of the way that those chromosomes and the information that they carry is organized as well. All right, so again, shortened to basically no interphase, but it's there. All right, remember again, there is no genetic duplication. Okay, so we're not going to copy the genetic information again, which is why it can be so much shorter. There really is no growth or anything like that either. Prophase 2, those chromosomes condense again, nuclear envelope breaks down, spindle fibers form. Metaphase 2, sister chromatids align along that metaphase plate. Anaphase 2, the sister chromatids split into individual daughter chromosomes. And then telophase 2 and cytokinesis, those nuclei form. And now I'm dealing with 4 haploid daughter cells at the end. So again, meiosis 2 looks a lot more like metaphase did, excuse me, mitosis did. but we don't have 46 chromosomes. I only have 23, and it's happening simultaneously in 2 cells instead of just one. Now, here's another interesting component to this. All of meiosis is meant to make sex cells, gametes, right? And we look at males versus females, and they each make different types of gametes. So the general principles of the stages of meiosis we just talked about occur the same way in males and females, right? In the production of that sperm and that production of those eggs or those ova, the processes of separating homologous chromosomes, separating sister chromatids happens the same way. What really does look different, however, is the cytokinesis, how we split the cytoplasm. So when I look at males, right, and their production of sperm, it is pretty much what we just said. I'm going to start off with a male germ cell up here, right? So that's basically what this is up here. These are your germ cells. Okay. And those germ cells will go through meiosis 1, meiosis 2, and in males, they'll produce 4 sperm. Okay, so it's equal division of the cytoplasm when I go through and produce sperm production, which is actually called spermatogenesis, right? Genesis meaning to create, spermata, sperm, creating sperm, right? In females, however, she goes through an unequal cytokinesis. So for males, it's 4 daughter cells at the end, just like we've been talking about. So 1 parent cell to 4 daughter cells, 4 sperm. For a female, however, which is what's over here, that oogenesis or ovum production, egg production, she goes through an unequal division of the cytoplasm. So she's going to take that initial parent germ cell, go through meiosis 1, create 2 cells. But look at the distribution of cytoplasm. This cell is much bigger. She goes through the second round of division, and again, there's an unequal distribution of cytoplasm. Her egg that she ultimately produces, or that ovum she produces, is much bigger than these 4 sperm here in terms of their cytoplasmic contents. And there's a reason for it. So she's going to be able to go from 1 germ cell to a single egg and these 3 things called polar bodies. All right. So males ultimately produce 4 sperm, right? Start off with 1 parent cell, males produce 4 sperm. Okay. Females, however, are going to go through this process and only produce 1 egg. The other 3 cells that she produces are these things called polar bodies. All right. Those polar bodies will eventually get reabsorbed by the body. They never get used to make babies, okay, to make offspring. There's a reason for this. So although they'll contain genetic information, they really don't contain anything else. They don't contain organelles. They don't contain other molecules that are necessary for that cell to function and survive. That unequal distribution is a result of who gives you your first cell, right? You as a baby, where did your first cell come from? It came from your mother, all right? So that ovum, once it fuses with an egg cell, once it fuses with a sperm cell, right? So when these guys come together, dad's going to drop off genetic information when he fertilizes this cell. Everything else in here is baby's first cell, okay? So dad's basically going to contribute genetic information. Mom is going to contribute the other half of that genetic information, but the rest of her egg cell gives you all of your mitochondria, all of your ribosomes, all of your endoplasmic reticulum, your Golgi apparatus, all the other proteins and other molecules that that cell needs in order to go through those initial rounds of cell division to start making a baby. All right. So dad's sperm really just needs genetic information, and they've got quite a copious amount of um, mitochondria and sugars to go through um, cellular respiration in order to swim. That's it. They don't have to be particularly large cells. But egg cell is going to not only give baby everything it needs in terms of the second half of the genetic information that dad can't provide, but then it gives everything else. All right. So while dad's sperm is going to end up determining baby's sex, right? Because dad is actually going to end up giving an X or a Y. Mom can only ever give an X. Okay. So dad's sperm is going to contribute either an X chromosome or a Y chromosome. That's going to determine baby's sex. Mom's going to contribute the X, right? And then whatever this becomes. If dad gives an X, baby's a girl. If dad gives a Y, baby's a boy. All right. But then mom is contributing everything else in terms of the functional components to that first. Other interesting little piece here is that males won't really make those sperm until they reach sexual maturity and can pretty much make sperm then for the rest of their life. Females, on the other hand, are actually born with all of these primary oocytes that she'll ever create, right? So her body already is born with all of the egg cells that her body will ever create. Now, they won't go through these rounds of meiosis until she hits sexual maturity, till you get to puberty. But that's the reason that females eventually go through menopause, is that her body basically runs out of functional egg cells. And so her body stops menstruating. And of course, that throws in all sorts of fun things for us as girls. Yay! All right. So just kind of an interesting thing, right? So males can pretty much make sperm forever, but females are already born with as many eggs as she'll ever produce.